Assalamu alaikum. This is Muhammad Hubab. I'm going to teach you microscope and its all types. So let's start. We use microscope for to see the uh, object with the magnification and resolution. So we will say that it's an instrument which is used to magnify the uh, image of object and to make object's image on the uh, lenses or on an device like camera, etc. So now we are going to talk about its parts. uh, these are some parts of microscope. Here I'm talking about compound microscope. Objective lens are of about there are three kind of objective lens with different magnification power: 40, 60, 80 or 100. Eyepiece are used to see the image which is formed from it. Human eye use it to see the object. And next one is the stage. The specimen is placed on the stage to see, and coarse adjustment is used to focus the object, and a fine adjustment does the same work. And next is mirror is used to throw light on the object so that we can see. Next one is arm is used to hold the object, a microscope. And next one is body tube. And mm, nose carries the objective lenses. Base is used to place the microscope anywhere we want. These are, I have said that these are all the parts of a compound microscope, because different microscopes have different types of parts to complete their requirements. Next, talk about their types. First of all, we are talking about the types of microscope by a source. There are there are three sources to see an object, like light microscope and electron microscope, and next one I think atomic absorption or atomic emission. So these are, and now I will talk about just two here: light microscope and electron microscope. Light is used to see the object. Similarly, our electron is used to make the object. If we talk about the types of light microscope, there are about four types of light microscope. First one is bright field microscope, and the next one is dark field microscope. We will explain it later. uh, like that, there is also a fluorescent microscope and polarized microscope. And now we are going to talk about the electron microscope. It also has I think two types. So first one is scanning electron microscope, and Next one is I'm providing you with all types of microscopes. You just have to study the electron microscope from other sources like websites, and you can use Google. I will just explain here light microscope, its types, and other types of microscope with other sources. Not means you can see there other types. We can also classify the microscope on the basis of type of number of lenses. So it is there are two types. First one is simple microscope having one lens. Similarly, compound microscope having two lenses. Simple microscope was used early and compound microscope are used now. Similarly, there is a stereo microscope and biological microscope. Microscope is used nowadays, and stereo microscope is also used to for the taxonomist to see the morphology of a large object like that, and like we use an Musca domestica, a bee, to see its morphology. We outside we can use stereo microscope. Similarly, next one is upright. In upright microscope, we will discuss this later. Just see the video. What is bright field microscope and light microscope? So light microscope is uh, let's talk about light microscope. It uses light, a visible light, white light, to see the image of object, to make the image of an object. So next one is bright field microscope. What is bright field microscope? Here's a stage of a microscope where an object is placed. Okay, and light will focus the whole stage, will it's a light source. The object placed on the stage will absorb the light, and the um, image formed will be dark. The dense area will be black, and in this way we can differentiate different things, uh, different contrast on the object which is absorbing the light, which is the absorbed remaining field will be bright, because light is going on all the stage, whole stage. So its disadvantage is what, that a transparent cell can't be seen. The reason is that light will pass through the object, and dense area is used to make black image, so it is the reason. Next one is dark field microscope. Also, I'm going to tell you that what is that. This is a object which is placed on the stage. Light will focus only on the object, and light will scatter this scattering light is used to make the image, and the other field is dark. The other field is dark. The reason is that light is focusing only on the object. Scattered light from the object is used to make the image. This is whole field is dark. In this way we can easily see the contrast. Let's talk about next type. It is fluorescent. In this type of microscope we dye the object with the color, and with fluorescent color, and then we pass a light. The light will emit the electrons from the object, and then will electrons will return back emitting light. This light is used to make the image, and this is we call a fluorescent my light microscope. Dye the object with the stain, and these are steps I'm going to tell you. And for these all purposes we use different parts, different components of microscopes. uh, and one thing I want to tell you that only dead cells can be seen, because on dyeing, on staining, the cell is dead. So we can't use a live cell by to see the live cell by fluorescent microscope. Is not possible. Passed electrons are passed, are emitted by the staining material, and then they return back. This is used in this way. We use this fluorescent microscope. Next one is polarized microscope. I'm just going to give you uh, general knowledge about them. You just have to if you want to detail of the all, you can search uh, them. You can search them separately. I actually collected the knowledge from different sources. I was just finding them separately. I then merged all these types, and and decided to make a video. Uh, Now all types are merged here. You can separately find their details from the websites. You can also use Google for searching websites. So next, polarized light. What is polarized light? In it we use light of two phases, 45 and 90 degree. In this way we are using two phases of light, two light phases, and we can easily find a structure, outside structure of a cell. It's all bumps which are easily made by two planes of light. Next one, we will talk about other types of microscope. I have told you that I will not discuss electron microscope or atomic. We are just here describing the light microscope and other types of microscopes. In simple microscope there will be one lens. These are were used early. Early scientists in 19th century used the simple microscope having just one lens. So this is light source at stage. This one is light source mirror. This one is a one lens, and this is stage. This was a simple microscope. Next we'll talk about compound microscope. It has two lenses. You can see that. Mm, one is an eyepiece. To see the object. To see the image, we use eyepiece. It also has lenses, and similarly in nose piece, it is objective lens. There are three types of objective lens we have discussed. This is body tube and arm, and this is stage, and this is last one is base. Next one is stereo microscope. It is used to see the 3D image, and scientists use it to see the large object like bee. Taxonomists used to see the morphology, like to see the wings of the object, and different legs. Biological microscope we use nowadays, all are biological microscopes. Similarly, upright microscope have what is upright microscope? Upright microscope have lenses above the from the upper side. From the upper side, we have lenses. In inverted microscope, we have lenses from below the specimen or object. You can see that open the stage has lenses below it. These are eyepieces to see the object from human eye, and these are objective lenses below the stage to see the object from lower side, from below. So eye, this one is eyepiece to see the object from the human eye.